Hello everyone, I'm Zhang from the Chinese University of Hong Kong. I'm very glad to share our latest paper published in Gut. Hepatic TM6SF2 activates antitumour immunity to suppress MASLD-related hepatocellular carcinoma and boost immunotherapy.
Metabolic dysfunction-associated steatotic liver disease is the most prevalent chronic liver disease globally. It can progress to steatohepatitis, fibrosis, and even hepatocellular carcinoma. Unfortunately, MASLD-HCC is resistant to immunotherapy and has limited therapeutic options. TM6F2 plays a protective role against the MASLD. However, its specific role and mechanism in MASLD-HCC remain unknown. In our study, we first analyzed TM6F2 expression in human MASLD-HCC tumors and adjacent normal tissues. We found that TM6F2 is significantly downregulated in primary MASLD-HCC tumors. To further explore the role of TM6F2 in MASLD-HCC, we established mice with hepatocyte-specific TM6F2 knockout and fed with high-fat high-cholesterol diet for 15 weeks. TM6F2 deficiency exacerbated diet-induced MASLD-HCC formation in mice.
The tumor-inhibiting role of TM6F2 in MASLD-HCC was confirmed in additional mouse models.
The next question is, what's the mechanism of TM6SF2 deficiency induced MASLD-HCC? To answer this question, we performed RNA sequencing on tumor tissues from TM6SF2 knockout and wild-type mice. We identified that immune-related pathways were enriched in TM6SF2 knockout tumors, suggesting that hepatic TM6SF2 deficiency reshaped the tumor immune microenvironment in MASLD-HCC. We therefore profiled immune cell populations in tumor tissues of orthotopic or transgenic mice. Impaired anti-tumor immunity was observed in mice with TM6SF2 knockout, as indicated by a marked decrease in CD8 positive T cells, especially IFN gamma positive CD8 positive T cells. Conversely, TM6SF2 overexpression in orthotopic MASLD-HCC mice boosted CD8 positive T cells and cytotoxic CD8 positive T cells activation. This leads us to another question: how does TM6SF2 affect cytotoxic CD8 positive T cells in MASLD-HCC? We examined the cytokine levels in the culture supernatants of human MASLD-HCC cell lines, and we found that interleukin-6 was significantly upregulated in the culture supernatants of MASLD-HCC cell lines with TM6SF2 knockout. Further validation confirmed that TM6SF2 regulates interleukin-6 secretion both in vitro and in vivo. We next investigate whether hepatocyte TM6SF2 could enhance cytotoxic CD8 positive T cells function by suppressing interleukin-6 secretion. The results show that the conditional media of the interleukin-6 antibody-treated HCC cells significantly abolished the reduction in CD8 positive T cells and the IFN gamma CD8 positive T cells induced by TM6SF2 knockout. In contrast, the conditional media of TM6SF2 overexpressing HCC cells significantly enhanced the CD8 positive T cells and IFN gamma CD8 positive T cells, whereas such effect was reversed by the addition of recombinant interleukin-6. This result indicated TM6SF2 modulates cytotoxic T cell function via interleukin-6. This raised a critical question: how does TM6SF2 regulate interleukin-6 secretion in MASLD-HCC? Next, we performed a pull-down assay and mass spectrometry, identifying IKK beta as a potential binding partner for TM6SF2. The interaction between TM6SF2 and IKK beta was validated by a co-IP assay.
Last but not least, introducing TM6F2 via adenovirus improved anti-PD-1 therapy efficacy. In summary, TM6F2 protects against the MASLD-HCC by enhancing anti-tumor immunity and shows promise for improving immunotherapy effectiveness. Thank you.